Hi, my name is Pimchanok, and today my talk is going to be about photoprotection of tropical seagrasses in the upper intertidal. So bring you back a little bit to this region. Okay, so this is going to be a summary of the works we have done over the past 3 years. Our lab is at Prince of Songkla University in the south of Thailand, and it's actually still very, very young. We don't work exclusively on seagrass, sorry, but we also work with mangroves and aquatic plants. But we do work exclusively on physiology with a focus on photosynthesis. We are also affiliated with a research center called Coastal Oceanography and Climate Change. And it's my two colleagues up there. It was founded 1 year ago with an aim to do multidisciplinary research, but with a common focus on the impacts of climate change on the coastal process, and that could be biological, chemical, or physical. Back to the talk. I guess you all have seen a seagrass meadow with large tidal variation. And these upper intertidal areas are particularly interesting for me because they are dealing with extreme environmental changes on a daily basis. This is one of my study sites, and this is me. So during low tide, you can see this large uncovered area. And when the tide recedes, the plants will be directly exposed to air, which then lead to desiccation. And if this low tide coincides with midday, then the plants will also have to deal with high irradiance and high temperature. This exposure can persist for hours. And in Thailand, in the upper intertidal areas, the common seagrasses are usually Halophila ovalis, our favorite, and Thalassia, hemprichii, and Cymodocea rotundata. So my question is very, very simple. How do these seagrasses cope with such a stressful condition? With a focus still on how they keep their photosynthesis intact and running. So this is a small overview. I promise it's very small overview of how plants protect their photosynthetic apparatus. So excess light can be dissipated from the light harvesting complex as heat by the first line of defense, the collectively called non-photochemical quenching. So from now on, I'll call NPQ. And this includes several components. One of those is the Sunfield cycle. And if it fails, then it may lead to a production of the toxic photoproducts, such as reactive oxygen species, or ROS. And if this is produced, then it needs to be detoxified by the second line of defense, which consists of enzymatic and non-enzymatic antioxidants. If the two of them fail, then that may lead, to a damage of the core protein of photosystem 2, the D1 protein, and a repair, which is essentially a replacement of damaged D1 by the newly synthesized D1, will play a critical role in keeping photosynthesis running and functioning. I would like to stress that all these mechanisms do not only play a role under excess light, but also under other conditions, which disturbs the balance between light use and light harvesting, or both. So our first results come from my student's work on desiccation. He's right here as well. He presented his poster yesterday, but on a different topic. So here we work on Thalassia, hemprichii, and Halophila ovalis. So we apply 90 minutes of desiccation simply by placing the seagrass to dry on moist sand. And then we rehydrate them with control, which is seawater, or the seawater containing inhibitor of NPQ, or the inhibitor of protein synthesis in cytoplasms or in chloroplasts. These figures show the recovery period after the desiccation. Okay, so this is rehydration already. So for Halophila ovalis, you see that inhibiting protein synthesis in chloroplasts by CMP, and inhibiting NPQ by BTT had really strong effect on Halophila ovalis, whereas inhibiting protein synthesis both in the chloroplast or in the cytoplasm did not stop, but slowed down the recovery of Thalassia hemprichii. So we concluded that NPQ and de novo protein synthesis had important role during the recovery from desiccation in the two seagrasses, but the extent depends on the species. Then my other students work on the same seagrass species, but on higher radiance, and this is published here in Botanica Marina special issue. And so we apply 3 hours of 3 light levels, and then we assess PAM fluorometer, we measure ROS, and we measure the activity of superoxide dismutase and ascorbate peroxidase. With limited time, I will only focus on the photoprotection part. So we see an increase in NPQ in Halophila ovalis under strong light, concomitantly with an increase in ascorbate peroxidase activity, but no change was observed in Thalassia hemprichii. So we concluded that Halophila ovalis is more physiologically responsive to higher radiance, and that NPQ and antioxidant enzyme are important for them to combat high light stress. What we additionally found is the clumping of chloroplasts. So under strong light, the leaves became pale, and under the microscope, we saw this clump of chloroplasts. And this has been reported in Halophila decipiens as well as Halophila stipulacea, as Sven just mentioned, under strong light and under high UV radiation. So this tight clump of chloroplasts, it has been proposed that it is a way to self-shade so that the light is less absorbed, and then therefore they get less stressed by high light intensity. In certain localities, red seagrasses are found, and this is due to accumulation of a non-photochemical pigment called anthocyanin. We worked with Halophila ovalis and Cymodocea rotundata, and we were very lucky to have the red and the green plants at the same elevation level and in very close proximity. So that allowed us to test our first question, is that does anthocyanin have photoprotective function? So we chose to do it by comparing the PAM parameters as well as the ROS production during the day. And the second question has been debated quite a lot in the terrestrial plant research, is that is anthocyanin is actually a compensation for lower capacity of other photoprotective systems, such as antioxidants or the Songkla cycle. So we compare these parameters between the red and the green plants. So the two tables summarize the whole paper, so it's a bit condensed. So for the first table to your left, so did red seagrasses do better under strong light? Here the PAM parameters suggest that they were doing equally well, except a bit for red Cymodocea rotundata, which produced slightly less ROS than the green Cymodocea rotundata. So, right now, so we say that anthocyanin perhaps did not really give that significant advantage to the seagrasses in our circumstances. So the next table, we compare this, all these photoprotection related parameters. So if anthocyanin is indeed a compensation for inferior photoprotective mechanisms, then the red seagrass should display a lower capacity for all these other photoprotection. It was not true in many of the parameters except for the Songkla cycle. In Cymodocea rotundata, both the pool and the deoxidation state. So then it is not really a compensation either. What we can conclude for now is that the red and the green seagrasses are using different combination of photoprotection, and that allowed them to cope equally well cope with high light equally well in situ in the upper intertidals. So from what we see in common, I still didn't get the first ding. Okay, so I was so afraid to overtime. So now I can slow down. So from what we see in common, this one thing at least I see in common is this non-photochemical quenching that keep on popping up in all the studies. And since it's also the first line of defense, and I am really highly attracted by it. And so now an ongoing work is going to be about characterizing non-photochemical quenching and its components. It's very nerdy, I know, but I like it. So you may have to take it 2 years after this in the presentation. And also, to properly answer ecophysiological questions, it's inevitable to consider combined stress. So particularly in the upper intertidal areas where I like, which, well, desiccation, high light, and high temperature could co-occur. So that would be our next step as well. And I'm trying to buy time here. So I would like to thank my funding agency and also my collaborators who really allow this so-called blue sky research to happen. And these are only under PSU, under Prince of Songkla University's roof. I see my collaborators here, both former collaborators and future collaborators and current collaborators. So I also thank you for past work and future work to come. While you're taking a look at this version of my floating PAM, I'll take questions. Thank you.
Thank you, Pimchanok. Really nice. I'm wondering if you've looked at the location, intracellular location of this anthocyanins.
I didn't personally. Anshana's group did, actually. So they did the section. So basically, well, they located in the epidermal cell largely. And from the references, it's mainly vacuolar pigment.
Yeah, but that would mean that at least part of the chloroplast would be above those anthocyanins, right? So,
well, I'm not sure, since plant vacuole is relatively big. Well, I'm not sure if it's the same case for seagrasses. So, well, if they're going to work as shading, I think they can.
Thank you. I shouldn't have sat in the middle.
Okay, one question from Svenby and another question from Peter.
No. Since you told us what you're going to do over the next 2 years, tell us what your hypothesis for the components is going to be. How do you think your MPQ of 7 is going to be broken down into components?
Yes, I'm toying with the inhibitors at the moment. So I'm trying to break it down into either, well, is it delta pH dependent? Is it all centrifugal cycle? My hope lies on the PSBS, but right now it's that hope is becoming more and more slim after I test with the inhibitor. Right. What I'm really interested in is the induction and the relaxation of it. Because, well, in my mind, I think like, okay, if they're dealing with this daily basis change, which is in a matter of hours, and then when the tide comes back, then, well, in some places it gets murky really fast. So, well, it has to induce NPQ really quick, as well as relax it really quick as well, compared to the other plants which are not dealing with this kind of problem. Yeah, we'll see in 2 years. Thank you.